I think we're live now. So thank you everyone for joining us today. This is our regular Friday seminar in the Open Source Neuro series, which is part of the Worldwide Science Initiative. And this series is all about open tools and methods in neuroscience that we collect in our website, openneuroscience.com. It's open-neuroscience.com. And you can upload projects to the website and sign up to give a talk like Lex did for presenting your projects using our form. It's very easy. You just sign up and we'll get in touch with you. And we, of course, welcome your contributions. And you can also share content with us on Twitter. You can find us there in @OpenNeurosci. And you will find some relevant links in the chat that we're going to put there in the YouTube. Today we have a personal favorite of mine, which is Dr. Lex Kravitz. Dr. Lex Kravitz is an associate professor in the psychiatry department at Washington University St. Louis. His lab seeks to understand how brain circuits control and guide behavior, with a particular focus on obesity and weight loss. He uses a range of behavioral and electrophysiological approaches in his research. And in addition, he has a strong interest in open source hardware for neuroscience research, co-founding openbehavior.org, which I encourage everyone to go check. It's a website with more than 200 open source projects, and he himself has released several open source devices for understanding mouse behavior, and Fed3 is one of them. He's going to talk about that today. In recognition of these efforts, he has received the NIDDK Innovation Award in 2016 and a Venture Funds Award from the Health and Human Services in 2017. Today, he's going to talk about a home cage device for behavioral neuroscience and introduce us to his creation, the Fed3. So without further ado, I give you all Dr. Lex Kravitz.
Cool. Thank you. Thank you so much for inviting me and for the wonderful introduction. So I'm going to talk today about a device. It's an open source device for training mice that we call Fed3. And I want to explain a little bit of the background, sort of why we made it, what was the purpose here, what kind of problem are we trying to solve, and then tell you about the device, show you some data, and kind of tell you how, if you want to play with the device or you want to use it in your own research, how to go about that. I should first mention I have no conflicts of interest. There's a company that's now selling the Fed3, and I'm very supportive of them, but I'm not financially involved with them. I'll mention them at the end. I think they're doing wonderful work for bringing open source tools to making them more available. And also, I realize this is being broadcast on YouTube, so this bit is a little bit of a moot point, but please feel free to screenshot and share anything. There's nothing proprietary or anything secret that I'm going to share here. And if you're sharing on Twitter, you can use the Open Neuroscience and Worldwide Neuro tags. So what is this device, Fed3? Here, looking at a photograph of it. It's a device, meaning that often behavioral systems are more of a system, a computer, a lot of wires, some hardware that all kind of interact. This is more, it's a device. It's a standalone, I wish I had one with me actually, but it's a standalone device that you can hold and place in a home cage. It's battery powered. One of the ideas of putting it in the home cage for mice was to have no wires, so we could really just drop it in and have it start interacting with the mouse, or the mouse start interacting with it. Almost like a little video game system you give the mouse, and then you can sort of collect data on the mice from it. So it's battery powered, it stores its own data. Its main application is that it's a pellet dispenser. So when the mouse does things correctly, it gets a pellet. And the way that the mouse interacts with it are through these two nose pokes. And we'll see some videos of mice interacting. So we can program it. So, poke on the left side and you get a pellet, or we can have something more complicated, like poke on the left side and then poke on the right side and get a pellet, or any combination like that. I mentioned that it's battery powered and it's home cage compatible. This is a photograph of it sitting in one of our home cages. So it's kind of small, doesn't take up a ton of space, and the idea being you can just drop it right in on the cage where the mouse lives. So why did we make this? And really the subtext, what problem were we trying to solve with creating a device? And my lab studies obesity in mice. Here we're seeing a photograph of, on the left, an obese mouse, on the right, a lean mouse. And really, there's a lot that's not known about both why groups of mice become obese and individual variability. I'll show you an example of a question that we've been working on for several years. And that is that, like people, mice gain and lose weight at different rates. So here we're looking at the average of 32 mice at their weight curves over 11 weeks. In the shaded area here in the middle, they're being put on a high-fat diet, and you can see their weight curves creep up the longer they're on that diet. And then when we take them off the diet, their weight curves stabilize, or even for some mice will start to drop. But this isn't the whole story. This is the average that we're looking at here. If we look at all the individuals, we'll see that mice gain weight at very different rates. These are inbred C57 black 6 mice, and I find it pretty fascinating that just like humans are more or less vulnerable to weight gain, even this inbred strain of mice, some of them are very vulnerable to weight gain when they see a high-fat diet, and others are almost completely resistant. So why is this? Why do some individuals gain more weight than others? And if we take sort of a system science approach to obesity, obesity is very complicated, and there are many contributors, many that we're very aware of, food consumption or physical activity, some that are out of our control, like societal influences, and then things that are individual to the animal. So the individual, here it's being described as individual psychology, or the biology, sort of the genetic makeup of the individual. All of these can contribute and ultimately determine how vulnerable an animal is to gaining weight. When I showed you that high-fat diet experiment, really what this is us modifying the food environment. We can take the food, the low-fat food that rodents usually eat in their colonies, and we can change it with a high-fat food. This is very much like maybe if you think about the food environment anywhere in the world in 1970, and then look at the food environment today, and just the food that's around has changed. And this is effective for looking at things like, it tells you something. It tells you that yes, the food environment is sufficient to cause weight gain on average, but we're missing a lot. Doesn't tell us why do some animals live in that environment and gain no weight. And some of these other aspects of this graph are probably the answer that would shed light onto why some individuals don't gain weight. So this was our main motivation in the beginning with designing a device that we could put in home cages to try to get some insight into not just food consumption, but maybe their patterns of intake, or what's being described here is their psychology towards food. So the approach we wanted to take was to uncover behavioral features that would bias animals towards weight gain. Could we find out that an animal that is highly motivated for food, or that is willing to exert huge amounts of effort to get food, is actually the animal that's most likely to gain weight? So why would we not use traditional behavioral tests? This is a slide from our Washington University Animal Behavioral Core. They offer a range of behavioral testing, and these are very sort of traditional behavioral tests with a long history and a lot that's known about both how to perform the test and the neural mechanisms of sort of supporting these behaviors. So why could we not use these tests to get at this big picture question? And the main reason is that these tests are often sort of time consuming to perform, and it's difficult to perform lots of them. So I'll give you an example. This was in 2017. We published a paper where we were looking at the interaction between activity levels and obesity. And we used a sort of traditional test to measure activity, which is just put a mouse in a box and use a video camera to track how much does this mouse move. And on this graph here, we're seeing lean mice in gray, and everything's been normalized to them. They're at 100%. And obese mice, as they keep gaining weight over 18 weeks, their activity levels drop and drop and drop. This is not a huge surprise. Something that was a little surprising to us at the time was that if we did a correlation and we ask, okay, so are the animals that are the least active, are those the ones that are going to be gaining the most weight? We saw no relationship. And this is something that's been replicated in many ways, that the relationship between, if I look at, say, this heaviest mouse over here on the right, let me see if I have over here on the right, this mouse, is kind of in the middle in terms of his activity levels. I'm looking on the left here. So this is okay. I think this is an okay approach for moving forward, but this took a lot of work to gain data from, in this case, just 8 mice. We've done larger groups, but pulling them out of their cages, they're pulled out every week, or sometimes we skipped a week, so every 2 weeks. We take out 16 mice, we put them on the floor, we videotape them, we process the videos. By the time you're done with all of this, you spent almost all of your time collecting data on a group of 16 mice, and you're the density of the data is pretty low. We're ultimately recording, in this experiment, we're recording about 20 minutes per week of the animals, which calculates out to about 1% of their sort of life that we actually managed to observe. So we started thinking that what we really need are methods for much higher power, being ways that we can measure activity or other behaviors around the clock at all times, have them be very little effort for the experimenter, so that if we want to get to 50 mice per group or 100 mice per group or test more than one form of diet, all these things that are just not possible, with a small lab kind of doing videotaping of these mice. So it took a bit of an inspiration from looking at other fields and how they've solved the same problem. So one of them in particular that's been very successful is the field of microbiology. Test tubes basically went out in the 1960s and in favor of well plates. And the reason is really obvious. A well plate is either 96 or 384 or even higher than that, individual reactions that can all happen at once, kind of in parallel, and essentially taking advantage of automation and just higher density to increase the throughput of the questions that can be asked. So if I look at this well plate and I think about our open field task that we did, it's a little bit like, we've filled in one well. And now if we want to go fill in the next well, we go back to our colony and we videotape another mouse. And slowly we can fill up a plate. But we might get to the end of a year and realize that we've only got one plate filled. We also started thinking more and more about the home cage. The similarity between these two photographs is hopefully obvious, but we started thinking, could the home cage be sort of like a 96 well plate for behavior? Could we have each cage be a reaction and have sensors in there that can really study an individual mouse? And then this would sort of be the vision of using the colony space as a way to do high density experiments. And many other labs have had this idea and have been starting to make progress on it. I think it's more of a vision at this point for all of us, that I think the vision is viable and we're getting there step by step. But there's been many sort of an interest in this in the last, say, 5 or 10 years of people making progress on home cage devices and approaches for studying mice, and showing many advantages, such that the throughput advantage that I'm mentioning here, but also the animals are less stressed. You don't have to worry about that you're moving them from their cage to another environment, possibly taking an elevator to another floor, all these things that kind of can impact the final results. So just leaving them in the colony and letting them go for a week or two is sort of, in my vision, a much preferred way to go. So to give you a bit of history on the Fed3 and where that fits into this vision, Katrina Nguyen was a biomedical engineer. She's now a PhD student at Carnegie Mellon, and she joined my lab as a postdoc in 2016. And she had a very innovative, or she is a very innovative person with sort of an engineering mind to this. And she thought, well, why don't we make a little device that we can measure food intake? So she spent about a year designing a 3D printed device that's controlled by an Arduino, and she called it Fed. This was sort of the original one. What this device did was drop a pellet and monitor its presence. So it would sort of sit there waiting for the animal to take the pellet. And when the animal would take the pellet, it would log that time that it took the pellet to an internal SD card and then repeat. So over time, you could build up records of the timestamp of every pellet that was taken. I'll show you a quick video. I have to turn my quick video of what this looks like. So this is a mouse. This is the original Fed 1, and the mouse takes a pellet, and the Fed dispenses another one, and over time he can keep taking pellets. So this is what the data from that would look like. Here we're looking in rows at individual mice. So this is 8 mice in the different colors, each mouse in one color. And this is data across 3 days. We can see that during the dark phase, the animals take more pellets. Not surprisingly, that's when they're doing about 75% of their eating occurs in the night. And then during the light phase, when the lights turn on in the room, their pellet taking drops, which is being plotted in average down on the bottom here. So this was pretty exciting to us, that we were kind of taking a step on our vision of, can we put something in the cage to start recording data around the clock? It's really getting at this food consumption part of this pie that I showed before. We started, for many reasons, we know that food consumption is just a piece of the pie. That won't tell you everything about why some animals gain weight more than others. There's also a lot of behaviors that are interesting to study that may be independent of weight gain and obesity. So we had a lot of interest in trying to kind of attack this other part of the pie, what's being called here individual psychology or animal behavioral traits. Things like animals' motivation or their persistence or willingness to work to get food. These were sort of the next frontier. And the postbac that started when Katrina left, Muhammad Ali, started working on FED and added a lot of the functionality, and kind of, I credit him with a lot of what FED has turned into. That a lot of these functionalities that we added really started with him kind of brainstorming and coming up with things. So probably the biggest addition that we added were these two nose pokes, which allow the animal to interact. So this is no longer a one-way street where the animal takes a pellet and the FED kind of just automatically does everything it does. Now we can program a little more interesting things into it because the animal doesn't just take pellets. He pokes on the left or pokes on the right, or we can come up with little games that he can play in order to get pellets. I have this photograph of Muhammad, and he's also in front of a poster he did. We spent a long time unsuccessfully, although I think it's still out there, trying to do group feeding devices. So if anyone has good eyes, you can see this is the open source group feeding experimentation device. And we were trying to use RFID tags and things, and I'd be happy in the question period to talk about some of the challenges we encountered. But I think this is an excellent future direction that we're sort of looking to get back into, maybe with some different technology, where instead of just one mouse interacting with the device, we could have a group of mice and keep track of which one's doing what. So I'll show you a quick video from Muhammad's device. This is now one of these sort of first generation with the nose pokes. But now the mouse can poke his nose and take a pellet. So that's about as simple a game as the mouse can play. And I kind of alluded to the fact that we can program it to do any sort of complicated game to kind of get at the learning of the mouse or its persistence or willingness to work to get pellets. One thing that came up, one of the challenges that came up, and I just want to take a minute to talk about it, is that whenever you have a pellet dispensing device, whether it's something open source or something commercial, it has the potential to jam. And it's just an annoying feature of things that move, that they may get stuck in some position. And so we came up with a lot of variants of how to dispense pellets, and ultimately came up with a unique sort of pellet dispensing mechanism that has been a huge help in reducing jams for these multi-day studies. So the mechanism, you're seeing the 3D design here and the pellet path in orange. The mechanism is that most pellet dispensers work by dispensing pellets at the bottom. You sort of have a hopper filled with pellets, and somewhere at the bottom, there's some type of mechanism for releasing a pellet. And we tried a lot of, this was sort of the original designs of FED, and it can work, but it's very susceptible that if a pellet gets in the wrong position, it can kind of gum up the whole works. So we came up with a different design where the pellets are actually being dispensed from the top. And there's a lot of sort of, I got this idea from watching videos on YouTube of different types of part feeders. If anybody's up late at night and wants to see a weird corner of the internet, you can look up how they sort bolts and how they sort different electrical components. And they have these really cool machines that vibrate and shake and will pull the parts up to a track. And so one of the mechanisms there was that I found very surprising at the time was to dispense them from the top. And what's nice about that is that then the pellet's kind of by itself, and the dispensing area stays clean because there's never a chance of too many of them sort of migrating up to the top. So how do we migrate the pellets up to the top and then drop them down? And the way we did this was with a sort of angled pellet dispenser. So I've just triggered it to a disc to start turning. And you'll see that the disc inside, instead of dispensing at the bottom, is pulling pellets up to the top. So here we're going to put some pellets into it. And you'll see what I mean. It's almost like a Ferris wheel. So that was FED detecting that a pellet was dispensed, and then it stops. These are some FEDs that were printed in a clear resin, and I'll just kind of show you the pellet path again, just because these videos came out super cool. Another aspect that we spent a long time with was trying to get the pellet to land directly inside of a sensing mechanism. So many pellet dispensers dispense a pellet, and it goes somewhere either into a little cup or maybe just into the cage. But it's not being monitored. And an important part of FED was that we want to know that the pellet is there, and because we want to know when the animal takes it. And this took a lot of iterations. This is what we came to. You can see it in the clear print. a mechanism where it's basically slowing the pellet down and getting it to land directly in there. I find these videos pretty cool. So as a result of these sort of pellet dispensings, we can use FED for long-term feeding studies. This is a 6-week study that we're actually still running now. But we're seeing 6 weeks of data, 1 week in the beginning, and then we started supplementing the animal's diet with high-fat diet in the middle, which is why the pellet counts on the Y go down so far. But the main point I wanted to make here was just that we're able to run these sort of in the home cage nonstop for 6 weeks, dispensing pellets. In addition, this mechanism, I've worked with a few other people, including Brian Swice at Mount Sinai School of Medicine, to adapt just this pellet dispensing mechanism out of FED3 and say, people want to use it as just a sort of simple, cheap 3D printed pellet dispensing mechanism. We put the designs online for this, and this is a task that they're running. You can kind of see it's really the bare bones of the FED3 pellet dispenser put in there. And they're running a task where they have 2 of these connected by a cable so that they can have an animal go on one side and get a pellet, and then the computer can know that he's already gotten a pellet on this side of the maze. Let's now give him a pellet on the other side of the maze, and kind of get the animal is induced to alternate back and forth to get pellets. We've also have a colleague that asked about using this pellet dispenser for human studies to get at food motivation in people doing eye tracking tasks, that sort of thing. This is Kyle Berger at University of North Carolina. And so we worked with him and designed a much bigger version of this that can be used by people. And because while we were designing it for him, we made one for ourselves as well. So we now have an M&M dispenser in our break room that's based on the same pellet dispensing mechanism. And if anyone likes this, these designs are also online. All right, so back to the mice. So what can this FED3 do? So the main advantage of putting it in the home cage, or there's several advantages, but one of them is that we can use it to automatically train mice. And what I mean by that is we can put it in the home cage, have animals be completely naive, and have them figure out what the game is. So this is that FR1, or 1 poke gets 1 pellet task, the simplest task we can do on this device. And we talked with 7 other research sites that are using it and compiled a group of data and basically asked people, can we have some of your data from the first time they've ever seen the FED? And we ended up getting data from 122 mice. What we can see is that at time 0 here on the x-axis, the animals are completely naive. They have 0 pokes because they've never sort of poked and gotten a FED. We're starting the experiment. And then over the first night of their training, the first 16 hours, we see they steadily increase pellets. If we look at poke efficiency, this is the % that they poke on the active side. So one side gives them a pellet, one side does not. See, in the first 4 hours, it's a little bit above 50%. Probably if we looked in the first 1 hour, it would be right at 50% while they're exploring. And then they gradually start to learn that only one side is giving them pellets. And over the first night, they get up to above 70% just from this simple task. Sinead Conway in Reem Al-Hassani's lab did a comparison here where she recorded kind of in a more traditional way using the FED3 in 1 hour daily sessions. So that's in these turquoise dots. And we looked at the same metric of poke efficiency. How are they learning which side gives them the pellet? We see that if you do 1 hour daily sessions, it takes about 20 hours or 20 sessions to get to 80% efficiency. If we do compare that to our overnight session, where they're getting there in the first night, it's pretty, it's a good demonstration of the power of running an overnight session, because we get basically all of their training done in the first night. It also tells you something about their learning, that it seems that the main determinant of their learning, at least on a simple task like this, is the amount of time they have to interact with it. So that time can be done in 1 hour sessions every day, or it can be done in 16 hours in one night. And the main advantage here is that it's not so much for this FR1 task, but if we want to do something more complicated, we've been playing with sequence tasks where they go left, right, left, that type of thing, which may be challenging or even too impossible to train them with short durations of time every day. But if we can give them, 24 hours, they can learn these more complicated things. Another feature I want to bring up is that because it's microprocessor because we control everything, we can synchronize it with external equipment. So this is an example of a self-stimulation experiment where we're expressing channelrhodopsin in D1 receptor expressing neurons in the dorsal striatum. These neurons are highly reinforcing. Animals will work to stimulate them. And this was collected by Alex Lagaria in my lab, but essentially just a simple, he pokes here, and instead of getting a pellet, we program the fed to pulse 20 hertz on a blue LED and give him light stimulation to the dorsal medial striatum. And we see that pretty rapidly, these animals will learn that to only poke on the active side. A really cool variant of this same approach is to take advantage of the fact that the fed knows when the animal takes a pellet. So now this was an experiment from the Crashes lab from Chris Mazzone and colleagues that was published last year in Nature Neuro. And what they used was they were stimulating the VTA dopamine neurons, so another population of highly reinforcing neurons. Animals will self-stimulate these neurons, but instead of using them as a self-stimulation, what they did was they paired it in a closed loop fashion with removing a pellet. So let me give my pointer back again. So the animals were fasted overnight, and then they were presented either with a fed without stimulation or with a fed that every time they take a pellet, they would get stimulation of the dopamine neurons. And what happened is that, not surprisingly, the animals take more pellets, but they actually consume more pellets if they supplement the removal of the pellet with dopamine stimulation. So Fed is saving all this data depending on how we run it. Especially, I showed you some data from 6 weeks of an experiment. You can imagine that this is a lot of nose pokes and a lot of data gets stored. This is an example from 1 week. I took a photo just because this mouse was so persistent. Mouse poking 24,000 times on the left nose poke over the course of a week. This was a progressive ratio session that I'll show you the behavioral paradigm in a few slides. But it took, it was poking this much to receive in the end 1,100 pellets over the week. But this generates a lot of data. And we sort of started drowning in the data of how do we understand this and the temporal relationships between how he's poking and how he's getting pellets. And Tom Ernest, who is a technician in my lab, who's now a PhD student at WashU, he made a Python-based graphical interface that's available online that was made for analyzing these data files. This is a screenshot of it, basically load the data in, and then it has many sort of pre-built analyses and data outputs that you can use for processing the files. And I'll show you a couple that are particularly useful. One of them is for analyzing meal patterns. So animals don't just take pellets on a completely regular basis. They tend to cluster them together in a term that can be thought of as meals, and then they take breaks in between the meals. And there's been a difficulty in the literature of understanding both what is a meal, what should classify as a meal, and we wanted to figure out if there was some sort of data-driven way, especially given that our pellets are 20 milligram sort of discrete units. It's kind of perfect for taking a data-driven way and saying, is there something in the pattern that we can understand, which may be challenging if we were to use, say, a dish of food and use video. And even though you could analyze his behavior, you may not know that sometimes he sits there for a minute and he eats very little, and sometimes he might sit there for a minute and eat quite a lot. But with these pellets, we have these sort of discrete units. So this is a snippet of the data file we're looking at. We see that every timestamp that's happened, every event that happens is timestamped. On the first column A, we have some information, and then in column F, I'm highlighting, it'll tell us what the event was. So a pellet was taken here on June 16th at 3:53 in the afternoon, and then the next pellet was taken at 3:54. If we plot a histogram of the distance between, or the duration between, each successive pellet, we get plots that look like this. I'm calling this an interpellet interval plot. What's really neat about these is it's a sort of data-driven way of understanding the relationship or the patterns that animals are eating. So on the x-axis here is a log scale of minutes between pellets. This is centered around 1 minute. And most animals will have a large peak around a minute or a little bit less than a minute, maybe 30 to 60 seconds. And these are these rapid fire pellets that when they get into a bout, they might eat 5 or 6 pellets in a row in short succession. And the distance between those, or the duration between those pellets, would be about 30 seconds to a minute. And that generates this large peak. There's a second peak that's out here, somewhere between 10 and 100, or sort of 1 to 2 hours. And these are the gaps in between meals. So animals will eat meals and then they'll break for 1 to 2 hours, and then they'll come back and they'll eat another meal and then break for 1 to 2 hours. And using this plot allows us to draw a line essentially and decide we're going to determine everything below a minute as a meal, or within a meal, everything above there we'll say is between meals. And then we can do analyses on these. And I don't so much want to draw attention to the specific results as much as just to kind of point out the things that are capable with this. In this case, we were comparing between dark and light. And we find is that the number of pellets per meal, or even the percentages of pellets in meals, are pretty similar between the dark cycle and the light cycle. The real difference is that in the dark cycle, they initiate more meals. So they just take more meals, but once they start taking meals, they're of similar length and the similar number of pellets. And that ends up accounting for why they end up eating more in the dark cycle when they're active. It's just they're initiating more meals. Tom came up with some cool visualizations I think are very insightful for gaining sort of understanding how animals are eating. So one of them is to understand the circadian rhythm. Because we're letting them go for a week or two in a lot of these experiments, we can get a very accurate estimate of where in the circadian cycle is the animal distributing its feeding or its operant behavior. This plot can be thought of like a 24-hour clock. So 0, the start of the light cycle is at the top, and then we go around the clock, and 12 hours in is at the bottom, or the end of the light cycle, and then the room lights turn off, and then this is the dark cycle. So this is a group of 8 animals, and we're seeing that you can just kind of get an idea of how many pellets they're taking in each of these bins, represented in red lines, and clearly they're eating more in the dark cycle. We can also do this plot in a different way, useful for comparing groups, where we're now plotting the mean of each animal with its own standard error. And in this case, group 1 and group 2 are not different, but we can sort of see the circadian cycle of their food intake and compare whether it changes between groups. So that's the end of sort of the data part I'm going to show you. I'm going to talk a little bit more about the open source nature of FED and how it's designed to be modified. Some of the modifications that are happening, there's been some interest in using it for rats. So Kevin Myers at Bucknell University sent me a photo, I guess this was about a year ago, showing some modifications to the design, trying to kind of make things bigger for use in rats. Sequin Wang in Shanghai has been doing much more extensive modifications, and they've been modifying FED3 largely with the goal of sort of simplifying manufacture or reducing the cost of manufacture. And so you get this kind of, I was delighted to see this sort of modification of our design with the same electronics that he sent me some photos of. In addition to the hardware itself, I should mention all the design files are online, including the editable, the 3D designs in the software they were created. So it kind of facilitates, or I'm certainly, hopefully facilitates people taking them and modifying them if they can sort of come up with any improvements. The other part of the open source is that the code to run it is all open source. And we've designed a library that runs in Arduino that makes behavioral programming, in my opinion, pretty easy on this device. So this is an example of a behavioral code. I'm not going to talk a lot through the code, but I will talk you through. This is the FR1 session, that simple behavior I kept saying, where they poke on the left, you get a pellet, poke on the right, nothing happens. And this is what it would take to program the FED to do that. And basically, I can talk you through. Line 24 says if the FED3 left, if a left poke happens, we're going to log it to the SD card, tell the SD card that a left poke happens. We'll play the conditioned stimulus. This is the tone and the lights, and then we'll drop a pellet. If a right poke happens, we'll just log it, but we're not going to do the stimulus and not drop a pellet. And this amount of programming, it will get the FED doing that task. And you can imagine how bringing math into this or something, you could easily say, next time around, now he's going to have to poke twice or come up with something more complicated. So in the last couple minutes of the talk, I'm going to get back to our original purpose. This has been a lot of fun, kind of creating a device and learning all these things and making this FED3. But our original goal here was to try to understand behavioral features that bias animals towards weight gain. So have we made any progress on that? And one task that we've been using that has been really helpful for studying mice and studying their motivation is a task that we adapted from the Beeler lab. You can see it published in this Mora et al. Publication from last year. And this is a closed economy progressive ratio task. What closed economy means, that term suggests that everything that the animal is eating is under operant control. So we don't have any free food in the task. That would be sort of an open economy where the animal's eating food that we're not sort of aware of. We're tracking every pellet, and it's earning every pellet by nose poking. Progressive ratio means that that the ratio of pokes that's required to get a pellet keeps progressively increasing. So the first pellet is 1 poke, the second pellet is 2 pokes, the third pellet is 3 pokes, kind of and on in that fashion. With one trick, which is that if we just left it like that, pretty soon the animal would have taken 60 pellets and he'd be up at hundreds of pokes required to get to the next one, and it just would end up not being able to get pellets. So in order to stop that, we have when he quits for 30 minutes, the ratio resets. So the animal can now take multiple strategies. If it wants, it can go really hard on the pokes and just hit high ratios, as this animal I'm showing here is getting. So here we're looking at 4 days of data, and we're seeing in orange the pokes, and in the purple trails, you're seeing each pellet and the number of pokes that were required to earn that pellet. So at its peak here, the one that I'm blowing out, this animal earned a pellet that required 84 pokes to get. So I had to go 84 times on that. But the trick is that if it decides to give up, if it quits poking altogether on both sides for 30 minutes, it goes all the way back to 1, and then it just continues. And we can run this for multiple days. And here the animals can take different strategies. If they want, they can just sort of take the easy ones. And then once it gets too hard, once it gets up to say FR8 or FR10, it can just quit and say, I'm going to just wait 30 minutes and then I'll get some easy pellets again. And then I'll wait 30 minutes. And that strategy is totally fine. It can obtain all the food it needs through that strategy. The term that's used kind of for an animal that adopts that is thriftiness. This would be an animal that's very efficient or very thrifty and using just the minimal amount of pokes it needs to get the food it needs. And the other strategy would be to sort of an anti-thrifty, just poke all the time and use a lot of energy, but you'll get your pellets by hitting these high ratios, like 80 pokes to get a pellet. So in this panel down here, we're just looking at body weights. This is, animals are able to obtain all their pellets through this task, given with evidence that they're not losing weight. And then we can plot these types of behaviors again across the circadian cycle, which I find these plots really useful. So we can see that they're obtaining their pellets. Something pretty interesting that we learned from this is that animals are really hitting their high ratios in the first 3 hours of the dark cycle. So now these plots are showing at 0 is the start of the dark cycle. And this is at 6 p.m. for our local time. And so between 6 and 9 p.m., they're really hitting their high ratios. And then by about midnight, they're back down and kind of finishing out the night. This is a different cycle than their activity cycle, which tends to be high for most of the night and then comes back down again. So finally, we want to see if this is sort of one feature of behavior that could we look at this, how thrifty or how anti-thrifty they are, and see if that correlates with the vulnerability. And what we found is that thriftiness, or that thrifty strategy, does correlate with a vulnerability to weight gain. So in this experiment, we had them running for a week on feds where we were characterizing this thriftiness. We were testing them the whole time through here with this progressive ratio, and then we put them on a high-fat diet. And our question is their thriftiness here related to their weight gain? And the answer was, I'll mention this is pretty preliminary, it's 7 animals, but really we saw a nice relationship where the animals that are most thrifty, that means that they're on the x-axis here, we're looking at the number of pokes they're willing to do per pellet. You can almost think of this in terms of economics, how much money are they willing to pay? They don't have money, but they have effort they can do. So this animal here that was sort of the most thrifty, or he would give up if the ratio got too high. Overall, he was averaging about 4 pokes per pellet, turned out to be our highest gainer. This animal over here, who's the least thrifty, he's willing to average about 11 pokes per pellet, is our lowest gainer. So I think this is a great example of sort of a behavioral home cage test that may give us more insight. And of course, obesity being multidimensional, we're thinking, beyond this sort of thriftiness. This may be one dimension, and I'm pretty shocked at how much variance is explained by it. But there may be, as we bring in 2 or 3 or 4 dimensions of their behavior towards food, we may be able to get a very good idea of which animal is vulnerable to becoming obese or not, which is sort of get back to our original question. So the last couple minutes, how can you use Fed3? If anyone's watching this and is interested in this device, I mentioned that that everything is open source and freely available online. The best place to start would be this GitHub link, which is Kravitz Lab Devices Fed3. And that includes information on the device, the electronics, the 3D design. There's also an Arduino programming library that can be downloaded through the Arduino IDE. And then finally, that analysis program that Tom Ernest made, FedViz. And all of these you can kind of navigate through. This would be the good starting point. And the last part, in addition to their sort of DIY instructions of build it yourself, Fed3 is being sold as an assembled thing or as a kit from the OpenEFIS production site. And I mentioned in the beginning that I don't have a financial relationship with them, but I'm very supportive. They're an organization that creates open source tools and fabricates them so that scientists can use them without having to build them themselves. They obviously started with their own OpenEFIS system, which is where their name came from, but they distribute miniscopes, they're distributing Fed3, they distribute pulse generators, several different open source tools. So they've been selling Fed3 for about a year and a half, and they've now distributed about 500 of these devices to researchers in over 18 countries. So if you're sort of interested in getting started and are not so interested in 3D printing and soldering and everything on your own, this is another resource for using Fed3. So I'm going to stop there. I want to acknowledge the people I called out. Katrina Nguyen and Muhammad Ali were instrumental in developing these first versions. Felipe Carjalvo is at OpenEFIS production site, and he's worked a lot. In addition to selling these, they've put a lot of effort into improving the design and working with me to kind of improve reliability and quality. And then I want to mention the other members of my lab who have all been contributing in various ways to the production and to the use of these. And with that, I'd be happy to take any questions.
Great. So that we leave a little bit of time for anybody who's on the stream, which is a little bit lag behind. I'm going to start the questions. Thank you for the talk. I'm very curious about the experience collaborating with different facilities, different universities, all using FED, and what's your experience and where you see this going? If everyone has the same device and kind of the same paradigm, do you see like a collaborative effort, like even a collaborative grant effort to do science across different labs?
That's a wonderful question. So Mattia's asking sort of, so I showed a little bit of data how we collaborated and got 7 labs to contribute data so that we could sort of make those graphs. And that was a great experience in the sense that everybody sort of saw the value in sharing data. There is a value that they're all being collected on the same device. And so there's some sort of standardization. It also brings up that, they're in different places, they're different ages, different sort of mouse populations. So you get the benefit of seeing heterogeneity and trying to understand if that's a feature or if that's an important feature. I see this happening going further, and you asked about future directions. I think one of the challenges of the Fed3 at the moment is that everything is stored locally on the device. So when we want to get the data off of it, we have to pull a card out of it and take the device. You can probably see where I'm going with this, but we've been experimenting a lot with wireless data transfer. And there's some challenges there especially to do it reliably with a lot of devices in one place. But I see that being the future, that if we can get data off of it in real time, and the technology is all there. I think it's mostly ironing out the nuts and bolts so that it actually works. But if we can get it wirelessly coming out, then it becomes really possible to collaborate, where we could sort of design an experiment together and have both research sites posting into the same database automatically. There's no need for me to email you and ask for you to upload your data or email it to me or something. It would just be sort of in real time posting. I see that as the biggest development that we're trying to work for. I kind of see that as being like Fed4 is going to be a wireless version that will, I will just say, I mean, the technology is totally there. We use wireless things all the time. There's nothing prohibiting it. A lot of the challenges that have been more on the backside on sort of receiving the data and creating an infrastructure that works reliably and makes sense for sort of containing the data. Sending data out of a device is pretty straightforward.
Nice,
Hi,
nice.
Lex. Nice talk. Actually, it was very convenient to understand from a person without any background from neuroscience. So that was nice. I just had simple question. Like, so you had mentioned about that you had jams through the pipe. The so I was just wondering if you had ever used like pressure kind of device to reduce these jams, as also kind of release those pellets in a tea fashion, something to keep up going. Is there anything you thought of?
Sorry, it broke up a little bit. Were you asking if we used a sort of pressure mechanism?
Yeah, yeah.
So
And
we've tried a
also
lot.
time dependent.
Yeah. We've tried other mechanisms. I don't think we've explored everything. So absolutely, this is a place where if anybody is using it or has an idea and or wants to either download the 3D files or talk to me about it and work together. I don't think we've fully solved this, but we've tried other mechanisms, and this is sort of the best one, partly because of its simplicity. And it kind of has the ability to fail. So when it pulls the pellet up to the top, if the pellet falls out, that's okay. The next pellet is right behind it. So it kind of has this sort of, I don't know, it's not super precise, but that ends up being a good thing with a 3D printed part that maybe doesn't come out so precise. So it doesn't have to be, it kind of has some forgiveness in it. But we've tried some other mechanisms, and this is the best one that we've come to so far.
Uh, I also wanted to ask about that external device you had used in your experiment. Was it to reduce this jam, or there was another purpose for it?
Oh, when I mentioned that we were connecting it to external devices.
Yeah.
So, no, that was mainly just to synchronize it with other data streams. So either to
Okay.
synchronize it with like a brain recording or with a brain stimulation.
Okay. I had another question. Like in the last slide, you mentioned about the relationship between thriftiness and then the weight gain. So, I mean, I was trying to understand the reason behind that. Like, is it like just because of the physical activity, or it's like more of a the gap period between your diet? Uh,
So it's, yeah, the questions about the thriftiness, this behavioral feature and whether it predicts vulnerability to weight gain. And it's not physical activity, but this idea of thriftiness is almost if some animals are very willing to expend energy, and they seem to believe they have unlimited amounts of it, and they sort of have a high energy phenotype that's not very efficient in the sense of if you were to think of how much energy is it taking to procure your food, it's not efficient, but it works well if the energy is available and they can use it. Other animals have a phenotype that is sort of for conserving energy, where they will do the sort of minimum work required to get the food they need. And that's described as a thrifty phenotype. And you can kind of see how this plays into obesity, that a phenotype that's trying to conserve energy may be more vulnerable to putting on to conserving fat stores when that fat stores are available or when the energy is available. And so that's sort of,
yeah. So that's what my question was. If someone is making more effort and doing more physical activity, then they should have less gain instead of more weight gain. But in your relationship, it was like kind of uh, you know the other way around. So that's my
I
question was
think
like,
that may have been a confusion in the way that I presented the graph, because it's exactly as you say, that the animals that are willing to expend more energy are the ones that were most resistant to weight gain. And the ones that were more thrifty and more conserving were the ones. So I think that may have been something confusing in the way I presented it, but it's the way that you're describing.
okay. One more thing. In the first slide, actually, you talked about the weight gain, like high fat diet. So I was wondering if you had the low fat diet as well as a control, or it was just mostly study with the high fat.
Yeah, we'll typically have the low fat diet, which does not produce obesity in mice for low
Okay.
fat.
Yeah. So yeah,
So
thank you for
let
answering my questions. Yeah.
Welcome.
I was wondering about the device you were telling us earlier about the material and manufacturing practices. So what kinds of materials were you or your collaborators testing out for these devices?
See, that's a great question. So we've been 3D printing this ourselves using not very expensive printers, so regular sort of filament-based methods. That's how we started. And we've gotten so people that have been using it have printed with a range of printers. We've become eventually we're sort of migrating towards using commercial services online that have much higher quality printers than we could afford. So these are usually SLS printers that make beautiful parts. I have like some parts here that are like, they're just like perfect kind of parts. But they have been printed in a range of printers and a range of sort of do-it-yourself to these high quality commercial printers. I will say that people that have, for each printer, when you download the parts, they will have different tolerance. And so people that were using, say, the resin printers from Formlabs, they ended up doing different tweaks to the designs to make it kind of get it dialed in for that specific printer. And this is sort of a challenge of 3D printing, that it's not as precise as machining or injection molding, where you are really down to like thousandths of an inch. And so it ends up, I would sort of say maybe a take home of, if somebody is downloading them, they may, if they have a printer that, we have a wiki where actually it has about 5 or 6 printers that people have used have been listed. But if you're using a different one, you probably are going to spend a little bit of time figuring out how to make it work on your printer.
Yeah, but since all the designs are open, then that can be done, right? So
yeah, absolutely. And something I've been wanting to do is actually start acquiring sort of like known code that works on each printer. So basically have a database of like, if you're using this model, you can download these files and it'll work. And this is just sort of in the back of my mind, and I've not done this yet.
Yeah, wonderful. Definitely can relate to the match your design to your printer part of it. I wanted to ask you, because you show something for rats, and potentially this can be open to different species, right? For example, Ceci works with birds. Would we have a bird feeding dispenser? Can you envision
Let's
4 or 5?
Yeah. I would say let's try it. I don't know if you'd have to tell me what, with they're eating, but I would think, I guess one is this, absolutely, let's try it. I'd be happy to try to invent something for birds. That sounds great. The other part is that the hardware itself and the control code and everything, that's the same. So even if you found that for birds, you need a completely different mechanism to dispense, I guess, something completely different, seeds. Even if you could still probably imagine the same control code, and maybe even the same, maybe they don't nose poke, but they may peck a key or something, and we could change the pokes for that. But I think in terms of that would be pretty trivial changes compared to the way it's operating behind the scenes.
Yeah, that's interesting. I was more interested in the dispensing part, because the other would be, I think, I guess, a singing stimulus for us, but for many people it would be pecking. We'd have to think about it. It's the little groups of seeds that would be weird, I think, to dispense, but
yeah,
interesting to think about it.
Okay, so I think we are done for the day. We can call it, and we thank you, Lex, for spending some time with us and having a wonderful presentation. Thank you for all your efforts that you do for the open source community. Again, we invite everyone to visit Open Behavior, and obviously to visit the different GitHub repositories where Lex has the many open source projects. And thank you for being here.
Yeah, thanks for having me.
All right. Bye.
Bye-bye.